ओके सर, अभी तो स्टार्ट है सर रिकॉर्डिंग ना.
Yes, yes.
Okay, good morning to all. And today the lecture series as per IQAC-ISF College of Pharmacy, which is going on. And this series, which is going very fruitful, because we are having a different kind of the speakers from the different areas of the world. So I welcome to the today's speaker Dr. Damanpreet Singh, who is now a scientist in CSIR-IHBT, and he did his M.Pharm from GNDU and PhD from PU Patiala University. He got so many awards, likely Dr. Hardyal Award, PC Dandiya Award, and a number of other awards he has got during his research tenure. And he also got a number of grants from DST, DBT, and CSIR. And he's currently working on a zebrafish targeting the area like epilepsy, seizures, and some other neurological disorders. So without taking much of time, I now request to director sir, please sir, say few words so that we can continue our talk.
Thank you very much, Dr. Samsher Singh ji. So, first of all, this is our moral duty to welcome our today's experts of this ISF dialogue series. He is a very dynamic, dedicated toward the research and profession, Dr. Damanpreet Singh ji. So, I welcome you from bottom of my heart. And I also welcome all these audience available on the online platform. So this is the excellent opportunity to all the audience and those who will gather connect after the lecture. It will be available on the YouTube channel. So I know that Dr. Damanpreet Singh ji is the very down to earth person and dedicated toward the research, and he is also very professional. So I hope this talk and his given the view related to the research and talk, it will be the very fruitful to each and everyone. So without taking time, I again welcome you, and I just give you the one information related to the ISF dialogue series. Purpose behind of this ISF dialogue series running by the IQAC team of ISF College of Pharmacy. So purpose is mainly of this one to interact and involve the young people, so that our young persons, young students, they will connect to each other. Let us consider I have a number of time phone call to you. Even that I am first time to face to you. Yes, this is the Dr. Damanpreet Singh ji. I am met you, but nowadays number of the person they can come to know that how we can connect to the peoples if they want to any professional you can say work related to the topic, they will connect to you. So this is the main motto, to provide the pure and justify information to the society. So again welcome you and thankful to you for accepting our invitation and provide your very precious time to deliver a lecture under the ISF dialogue series. Thank you very much, and over to Dr. Samsirji.
Thank you sir, thank you for your valuable words. So I request to Dr. Damanpreet sir, who is going to deliver a talk on multi-omics networking approach in drug discovery. Sir, I welcome you again and thank you for accepting our invitation. Please, you can continue your talk.
Thank you, Dr. Shamsher. And firstly, thank to Dr. Gupta for his nice and kind words. And at the same time, I also thank Gargji, again Dr. Gupta ji, and Dr. Narang and my friend Dr. Shamsher for inviting me and giving me this platform to interact with you all. Because I just read the name on my screen, and sometime I also get an opportunity to interact with some students of ISF college, and I'm always behind the behind seeing the activities of the college on their social site platform, either it's Facebook or on its Twitter. So, Dr. Shamsher, if you permit, can I share my slides?
Yes, you can share, sir.
Doctor, is it visible?
Yes, sir. Few seconds. I think so. It will take a time to upload.
Okay, okay. So, Dr. Shamsher, is it visible?
Yes, sir. Few second. I think so. It will take a time to upload.
Okay, okay. So, Dr. Shamsher, if you permit, can I share my slides?
Dr. Damanji, you are presenting your screen, so you can open your presentation on the window and small
Okay.
it, and then you can share.
Okay. The noise, is it visible?
अभी सर
Dr. Saab, I'll reconnect. I'll reconnect.
ओके ओके सर ओके अभी सर एक बार अपनी पीपीटी को एक बार क्लोज करके ओपन करें फिर देन यू कैन शेयर
Oh, no. Okay. Now, okay. So change your slide, please, Damanji.
यस नाउ विजिबल सर इट इज फाइन
Uh, Dr. Damanji, unmute, please. You are mute now.
सर यू हैव म्यूटेड योर वॉइस प्लीज अनम्यूट दिस स्पीकर
Now fine.
यस यस यस
Now, okay. Now, okay.
Okay. I apologize for the interruption again. So now coming to my presentation. So Dr. Shamsher, can I start?
यस यस वेलकम सर यू कैन कंटिन्यू
Okay. So this is just an view of our institute, our lab, and next that is just a small association of IHBT with ISF Moga. I think you can see the logo of ISFCP on the lab coat. So this is a photograph when your student with one faculty member visited our lab. I tried my best to showcase them the information that I can within this short span of time. This is just a small interaction. So as far as the institute it is concerned, you are doing excellent work, outstanding work in the field of pharmacology, where majority of the people that are working at ISF College, they are working on drug discovery and target validation. So, as the name it indicates, multi-omics. As Dr. Shamsher has already told me that it is a part of their M-Form curriculum also. So its name is big. It is multi-omics. So omics. Now we see a lot of things like foodomics, again Ayur genomics. So these are some omics. When we say it is a big data, so we associate something with big data. So how this drug discovery and target validation now we are doing in a traditional manner, like we have some information available on literature that, like particularly you are making a drug for Alzheimer disease, so what is the pathway of Alzheimer's. So we just select the target on our own choice, taking into consideration about its specificity to particular disease, and then we find out several drugs and molecules, and we try to target that drug to that particular target. So this is just a selective target based drug discovery. But now we see that things have been changed. What we think, it is not exactly what is actually happening actually. So my topic, it is related to that, how we can find out the drugs. I have not much more focused my this lecture to the theoretical knowledge. I have just tried to compile some practical examples where we can use this multi-omics approach and how it is being used. Its name is big, but when we see its application, it is very simple and fruitful. So just to give a brief background about what is omics, what are transcriptomics, what are these terms, I will just dedicate 3 slides, and later on I will just move to the examples. So it is omics, it is a collective characterization, quantification of pool of biological molecules. So it is just characterization of the pool of biological molecules or a chemical molecules. And the type of molecule that we are making pool or we are quantifying, we term that particular thing to related to that. Like if it is gene, then we say it is genomics. If it is protein, we say it is a proteomics. If it is a metabolite from a plant, we say it is a metabolomics. So its genomics, its term was described in 1990s, and after that, a lot of things has been happened to these particular genomic tools. So these are some of the terms that we use. It is your metabolomics, we use metagenomics, and many more nowadays. So simply what happened, how we perform this experiment, and how it can be applied in drug discovery. Like you have a particular disease brain. Like I have talked about Alzheimer. Samely, we are taking Alzheimer as an example. And for example, a person have died that was suffering from Alzheimer, died because of some other reason. We just take a part of the brain that was affected, and we just isolate the whole mRNA. And then we convert that mRNA into cDNA, and then we just make an array. Array is simply, we say it is a big term. It is simply a 96 well plate, so you can assume it. And in that each well we put a particular sample, and against that particular sample we add particular gene in each and every well. And there is a color that tell us that expression has been happened or not. By having this data set, we can able to know that which type of the genes are overexpressing, which type of the gene they are underexpressing. And then we make some network, and we find out which can be the potential reason of that particular disease that is being happening. And then we discover, or then we target our drug to that particular gene or particular protein. So simply we say, nowadays this omics, it has been, the application has been increased multifold because of recent growth in molecular biology, because earlier the data was not available. We don't have so much of platforms from where we can get the data set. But now with the advancement of molecular biology, everything is in public domain. There are a lot of platforms that we can use. Now we have computer. We say in 20-25 years from now back, there were no much more availability of computer and internet. That's why the target based drug discovery was just focused on the selective drug target discovery. So I will just take an example of diabetes. So you can see that diabetes mellitus, its disease, it was described in 1675. We say around 600 to 700 years back. Means after before 700 years back, we were aware of what is diabetes mellitus, but its first therapy was coined in 1921 by Frederick Banting. He got the Nobel Prize. He was working in University of Toronto, and he discovered the insulin, that this was a peptide that is present inside the body that reduces the insulin level, and that this can be a drug therapy for diabetes. So you can see the disease, it was termed in 1675, and the first therapy it was coined in after 500 to 550 years after the discovery of this disease. And if we say about the drugs, the oral hypoglycemics, the discovery started in 1900 when the world was involved in the First World War. So we get sulfonyl drugs during the World War II. And we say then there was a first line therapy with metformin for type 2 diabetes. We say we get the therapy after 500 to 600 years back, years after the discovery of a particular disease. So means it take a lot of time. And now we are at the point that we have made available a tablet of rupee 1. We have an anti-diabetic drug with the cost of 1 rupees. So now we can say that now we are having a complete knowledge and complete therapy of this particular disease. I'm just taking the example of diabetes because you are aware that majority of the population is suffering from this disease, and we assume that we know much more about diabetes, but the exact thing is not like what we see in this literature. If we see what is happening in past 2 or 3 years, so you can see the transcriptome sequence of peripheral blood of type 2 diabetes mellitus of the patient has been done. This indicates that the knowledge about the gene changes that has been happening in the diabetic patient, we were not aware before 2020. So means there are several genes that we were not aware of that are playing the role. May or may not we are targeting on insulin, that is a major target or major therapy for this disease. Similarly, we say the integrative transcriptome of the pancreatic cells, because what we assume that the diabetes, it happens when the pancreatic cells they get damaged, or there is we say some receptor problem of this insulin receptor that results in diabetes. But the transcriptome, what is happening inside the pancreas, was done 2 years back. So means we are getting the information about the diabetes in past 2 or 3 years. Similarly, transcriptome of the type 2 diabetic and healthy human neutrophil was done 2 years back. This means the information that has been extracted from these studies can be used for the discovery of better, safe drugs for the future. This is the opportunity that this omics approach it provide to us. So this is what it is happening inside, or we say it is happening in the drug discovery or molecular biology throughout the world. Now I just want to give example from our lab that how this transcriptome or this metagenome or we say genomics can help in drug discovery. Like I'm also from the pharma background. For us, like you all, this omics, it is something we say it is just a theoretical information. How we can use, how we can perform. We are not very much familiar of these things. The same was with me that I was not very much familiar with all these things. But luckily, I was working in one of the topmost institute that has a strong genomic background. We have PEC Bio, we have Illumina, that is really present in the institute that are present in the country. But we are lucky that we have all these this infrastructure in place in our institute. So I'll just give an example of epilepsy, as what Dr. Shamsher said, that our lab is working in the field of epilepsy. So I'll just give the example how we have applied this, and we have come out with some fruitful things that can be utilized in future for targeting a drug therapy. So this work, it was initiated by one of my student, that was Arindam. After he completed his PhD, he worked as a postdoctoral fellow at University of Rockstar, and presently he is working in Baylor College of Medicine, US. So his work, it was a similarly that I have told that what we do, we just pick up the target and we start working on that particular target. And since I had just joined this institute, I was having the same knowledge. We have to just pick up the target. So I took the target that was mammalian target of rapamycin, because this Arindam, he worked on epilepsy. And if we say what happened in epilepsy, it is an abnormal growth. What we know till now that epilepsy, it happens when there is an altered excitation or inhibition inside the brain. But if we see more logically, there are some abnormal structure that developed during epilepsy that results in appearance of spontaneous recurrent seizures. So his Arindam, he worked on inhibiting these work on inhibiting the abnormal growth that is happening inside the neuron. For that, we targeted this mammalian target of rapamycin. It was just a pick based on the literature. So when he started working, I think you all work on staining, or we say on imaging. And to have a better image inside the brain, we have to go for transcardial perfusion, in which we perfuse the whole body with a perfusate or a fixative solution, so that the whole tissue inside the body, they get fixed. And in this case, we were working to see the mossy fiber sprouting that happen inside the brain, and that is an end result of mTOR activation. So we were not interested into going for transcriptome and all these things. So one day what happened, Arindam, he came to me that, sir, जब हम in transcardial perfusion देते हैं heart से, in which we have to inject a perfusate solution in the ventricle, and then we have to cut the auricle so that the whole blood it get exited from the body, and in replacement we add the fixative solution inside the whole body vascular system. सो ही केम टू मी दैट कि सर जब एपिलेप्टिक एनिमल में देते हैं तो इजीली हो जाता है, लेकिन नॉर्मल में थोड़ी दिक्कत आती है. सो आई सेड यू माइट बी सेइंग डिफरेंटली. नॉर्मल में हो सकता है कि इट इज इजी एंड इट इज डिफिकल्ट इनसाइड द डिजीज्ड एनिमल हार्ट, बट ही सेड नो, ऐसा नहीं है सर, एपिलेप्टिक में इजीली जाता है. सो देन आई आस्क्ड हिम टू रिमूव द हार्ट फ्रॉम द बोथ एनिमल्स. सो दिस वाज द एंड रिजल्ट. यू कैन सी द एपिलेप्टिक हार्ट, इसका साइज जो है इट इज लिटिल बिट मोर, but for the normal nave it was little bit less. So because of this increased size of the heart, it was easy to give an injection inside the ventricular region. Basically it was an abnormality, that is a dilated cardiomyopathy. So ventricle was dilated, so it was easy to inject something inside the dilated tissue, but it was an abnormality. Then we tried ki what is happening. So when the next study we planned, in addition to what we were planning, we just note down the cardiovascular function that happened during the process of epileptogenesis. So it was really interesting that what we observed when we induces the injury inside the brain, and after there was a latent period and the time when the epileptic seizures started to appear, we note down the this arterial pressure, this MAP, it is a mean arterial pressure. So you can see during the latent phase, means after the injury and during there is no seizure, there is a decrease in pressure, and when the seizure started, the pressure it get increased and increased, and it further worsened when the duration of seizure it get increased. So that clearly indicated that because of this increased pressure, this ventricular region it get dilated. And when we note down the weight, obviously it was more because some inflammatory changes it was happening inside the heart. At the same time, we note down the QTC interval, it was increasing. When the duration of seizure it get increased, but still the things were not clear. So then we studied the literature. Is there any condition in which the heart is affected in case of epilepsy? So what we observed, it was really shocking. We found that in epilepsy patient, there is a higher risk of mortality compared to normal population. And some little bit literature was available that indicate that recurrent seizure after neuronal membranal function result in irregular transmission, and because of this or altered neurotransmission, particularly in autonomic nervous system, it was leading to this altered cardiac functioning. So when we found out the literature, it was interestingly to know that most of the epilepsy death have unknown cause with a little or no postmortem revolution. So that indicates that there is some death, and that death is having no underlying cause. The term it was given as sudden unexpected death in epilepsy. So this gives us a good edge to work in this condition, that if we can provide a therapy to a epileptic patient, in addition to cure for epilepsy, in which the death, we can avoid the death of a patient, it is more blessing from those patients. So since I said that I was from the pharma background, I have just picked up the target and just reported that information. So you can see I just carried out the histopathology. It was dilated cardiomyopathy. The cardiomyocyte thickness, it was clearly evident. This deposition of fibrous tissue, again it indicated the inflammatory changes, because of which we were getting the increased heart rate. And these were some of the selective protein that we have studied, and we have just reported this information. But still something it picked in the brain, that in my own brain, that something was happening that can be explored further. But still I was having a limited information or limited knowledge in this condition. After that, I just collaborated with one of my colleague. He was trained at CDFDA at Hyderabad, having a great knowledge of this genomic tools. So he said, rather than having this uh, selective expression studies, we should go for the complete transcriptome and proteome, so that we can see what is happening inside the whole genomic makeup of that heart of epileptic animal, and what are those genomic changes are leading to the altered translational that is happening at the proteome level. So thereafter we extended this study now with the focused target of this identifying the genes that are involved in the whole abnormality. We use lithium pilocarpine model. We make alumina. We use alumina library preparation and sequencing using Novasec 6000. This is a unique facility that is available in our institute. Then we studied the whole transcriptome, then differential gene expression. Just the names are big, but the end, what is happening, it is very simple. This differential gene expression is that this is a basal value. What are the genes that get up or down from this basal value? We call it as differential gene expression. Then we make some, we use an this in silico tool to make a pathway that how these genes are interacting to get the pathway that are involved. Then we studied the protein-protein interaction using this network analysis. And then we validated the whole gene and protein expressions with the wet lab analysis using RT-PCR and this LCMS, MS, QTOF, and Western blotting. So interestingly, when we studied the whole genomic makeup, the whole alteration in the genes that are in playing role in this, interestingly, or if you say it is shockingly, we found 1157 differentially expressed genes. But when we go for the selective, we just select one or two. May or may they are playing a crucial role or not. But here we found that there was 1157 differentially expressed genes. Means these are the genes that get altered. And making a drug therapy for all these genes, it is not practically possible. So thereafter, there was a need to go for studying the pathway analysis, that how these 1157 genes, they are making a pathway, and that crucial pathway that is involved in that particular condition. And this is how we make a set of those genes using Metascape. And then we found that all these this 1157 genes, they make all these 23 or 25 pathways that play a role in development of that particular condition, because of which there is first abnormality in cardiovascular function, and ultimately there is a death in epileptic patient. So after that, we make some network, again using an in silico tool. So you can see there are some intramodular and intermodular hubs. In naam hi bada hai, but basically what these intramodular and intermodular, these are just, we say these are the target in that particular pathway. We say if we remove this particular target, the whole pathway get disrupted, and that abnormality will not happen. So means if we have to target something, we have to target these particular crucial genes, because these are the genes that has stabilized that abnormal pathway that is happening inside the heart. So thereafter, we studied the whole proteome, because it is not always that what is happening, that it is not always that any gene that has been expressed will translate into a protein. Sometimes it is not like this. Sometimes the gene get expressed, but the protein get downregulated, because there are multiple mechanisms that are involved. But however, we first studied the proteome, and then we merge this proteomic data with the genomic data. Means what is happening at transcriptional level and what is happening at translational level, to find out, because ultimately what thing has to be done, it is to be done by the signaling. That signaling has to be brought by the proteins. And what is happening at the gene level, it has to be get translated. We were just interested to see how many genes they are getting translated into the proteins. So here we found that there was 1264 genes were getting differentially expressed, and then we merged the data of 1157 gene with the 1264 proteins. And after merging, you can see this blue, all these genes, these were the crucial intra or intermodular hubs that I have shown. that these are the genes that have stabilized those pathway. And among these genes, these are the genes that has been expressed at protein level, and the pink color, it indicates only those genes that has been overexpressed in both conditions. We means we can target all these genes, or we have to find out a pathway where all these genes are making an end product that is resulting to a particular disease. So to find out, we have carried out the pathway analysis, and then we validated these results with our gene QRT-PCR analysis, just to see that what exactly it is happening at transcription level is well, is truly we are getting at wet lab level or not. So it was in line, but in one or two cases, we get the discrepancies. Like you see SRC. In case of epileptic animal, it was totally abolished. So based on these genes, we then studied the protein at using Western blotting, and again we found that what we observed, that what the arrhythm he observed, it was in line. The treated patient, that was a treated animal that was treated with an mTOR inhibitor, their heart was maintained, and that clearly indicate this can be a potential target to find out a therapy that can be used in patient with a with a dual objective, that this particular pathway is playing role in epilepsy as well as it is playing role in epilepsy associated cardiac abnormalities. So we further want to explore this target, and interestingly this paper, it was published in Epilepsia. You see in this particular journal, the input from our country, it is very less, because it is one of the prominent journal based on of this International League Against Epilepsy. So their whole aim is that we are not focused on just preclinical study. They just want the thing that has to be published. They has to have some clinical relevance. So after the publication of this paper, it was cited very well in at good platforms. But then we just wanted to work further, that whatever we found, we just want to validate it. But for the validation, the problem was the specific inhibitor of mTOR, it was very costly. It cost was around 80 to 90 lakh if we have to use the whole substance for an animal study, because it was around 2 to 2.5 months study. So therefore, we have to find out some alternate. So thereafter, we developed a new model, particularly that was similar to the model that we have used in our previous study, where we use rat, but here we want to use zebrafish, because the dosage required for zebrafish, it is very less. The weight of animal in that case, it was 300 gram, but here it was 200 to 250 mg. So obviously the dose was less. It was in case of rat, the study was of 2 months, but here it was just for 28 days. So then we developed this model, and then we tested that particular mTOR inhibitor in this model. Luckily, the finding that we get at our omics level, at proteome and genome level, it was in line when we carried out everything at wet lab level. When we inhibited this mTOR with specific inhibitor, the abnormality that we get in zebrafish heart, it was not as it was there in the untreated fish. So the thing was properly validated, but still our institute, it is working on natural product. You can see the name of our institute, Institute of Himalayan Bioresource Technology. Then our head he said that you have a good lead, why don't you use all the molecules that we have in our institute to target this condition, that can we translate something, because one of our major aim of our institute is to have something that has some translational aspect, that we can give to the society or to the industry or to the market. So in our institute, we have a complete set of library of all those compounds that we have isolated, because we have a strong chemistry group, natural product chemistry. They isolate lot of molecule from these Himalayan plants, and using some in silico tool, because we have a protein now and we know what thing we need. We just targeted all the molecules that we have in our institute with that particular target virtually, and we found out that Nardostrichus datamansi, because this is a plant that is used for cardiovascular condition, as well as it has been reported to be used in Ayurveda for the treatment of epilepsy itself. our institute, one of our lab, it is working on developing plant tissue culture of this Nardostritis jetamansi, because it is a rare plant. We say it is difficult to find out in India, because there are a lot of other plants that are being used instead of Nardostritis jetamansi. Like one is Selenium vaginatum. People are using that plant in case of this, but still in our institute we have a good plant tissue culture, and from this we have established this particular plant. And we use this plant because all the compound that has been reported in this, they were fitting very good inside the protein that we have identified. So first of all, we extracted this, and then after extraction we carried out qt of ms to find out whether the compound they are present in that extract that are targeting this. And thereafter identification of all these compound, we targeted to the pathway of our interest, and it was fit, all these molecules, they were fitting very well into this. And thereafter we repeated the whole study, in this case in rat, because the amount of extract was not a problem in this case. In this case we used the rat model, where we induced epilepsy, and then we kept the animal to show recurrent seizure. And after that we removed the heart, and we carried out western blotting and gene expression analysis, and it showed that this plant, it has some good potential to be used for this condition, because it targeted all both the condition epilepsy and this epilepsy associated cardiac irregularities, having its good potential to be developed further. And we are working to further proceed this study in association with our clinical partners. So thank you, because I was just allotted 25-30 minutes, and I have just summarized our own examples. Thank you. So these are just
Thank
my communication details. If anyone who or she want to have some more knowledge in this case, I will love to share the information.
Thank you, sir. Thank you. It was a very good talk that you have discussed all the aspects at the molecular level. So if there is any audience who want to ask any type of question, because sir is working at the molecular level. So if you have any question, you are welcome to ask.
So, Dr. Shamsher, if you say, can I stop sharing? Because I am not
Yes,
able to.
yes, sir. Yes, yes. Yes,
Okay,
sir.
thank you. Thank you. So if any query at this moment, I will be very happy to address. But if anybody want to communicate with me later, I will be very happy to share the information.
To the cardiac remodeling and some cardiac changes that I seen that you are using so many, target based studies. So I will just ask a one question that whenever you go for the genomic proteomic, that is there any kind of gene particularly you are targeting, or you are found a new thing which is a newness for in the research area?
Uh, Very good question, Dr. Shamsher. Actually, we have just carried out this genomic tool for this particular condition, that is epilepsy-associated cardiac abnormality. And all the genes that we find out, they all were novel, because this condition, it is very rarely described. That I have already shown that in even post-mortem patients, there is no underlying cause of death. And definitely, what you are saying,
Okay. So
we can extend this study for cardiac remodeling or other models for this, maybe of this ischemia or all these things. It's definitely you will get a lot of new set of genes. Like you see, we have got
and
around 1100 genes. Definitely, I'm sure that if we carry out the conventional model, we will get a lot of more information that can be extended further.
And the second thing, whenever we go from the small species to the large species, the genomic variability will be there. So how
Yes.
you cope up with this type of problem?
Dr. Saab, what we did at this moment, it is that rat model.
Okay.
It is obvious that when we change the model, the things will get changed, but we say the whole pathogenic mechanism will remain same. Like what you said, like I have set mammalian target of rapamycin, but when we did it in fish, It is not a mammalian model. It is a non-mammalian model.
No,
It's
just, my question is, sir, suppose we know that as per the literature availability that the fish is having a good, regeneration power of the
yes,
heart,
true,
which is
true,
which is not comparable with the other animals.
true.
So, is there any such type of thing that you found there that there was a variability?
Yes, like what I explained, that we developed a new model of chronic epilepsy, because in chronic
Okay.
condition, the developing of this model, it is difficult because of this regeneration capacity. So our main focus was to develop such a model in which we don't give much more time for regeneration. Like for cardiac condition, you make cardiac injury today, its regeneration will start after 3 days. So we haven't give them much more time to for this regeneration. We completed the whole experiment because this was for 28 days exposure. We give injurious stimulus for continuously 28 days, and then we completed the study within 24 hours of that time span. The things are in our mind that it has a regeneration capacity, but there is a time gap of 3 days that we can utilize.
Okay, sir. Thank you. It was a very good lecture, and I hope that all the students they got a lot of knowledge. And even though sir had share the Gmail as well as some other contact, you can also contact him. And now I request to all the student and all the the faculty member, please you can exit so that we can just talk with Dr. Sir for a 2 minute. So please जितने भी student है आप exit कर लें.
So I think now you can stop recording also. The lecture is over.
Yes, yes, sir. Thank you.